that this webinar today is the third webinar in a series in which we cover various aspects along the workflow for microbial genomics, or in this case microbiome research. QIAGEN strives to provide researchers with the solutions for the problems that occur along all steps of research workflows, such as microbiome research. In the first webinar covered the processing of complex microbiota samples and how we can help researchers address problems such as extraction bias or the removal of inhibitors. The second webinar followed up on NGS library preparation and introduced you to methods that allow you to produce high quality and low bias libraries for next generation sequencing. And today we'll focus on the last step in this process, on the analysis of microbiome data and how to glean insights into the biology after the analysis is completed. On this slide, I'll quickly show you or walk you through the agenda for today's presentation. First, I'll introduce you to the Microbial Genomics Pro Suite bundle. This is a product bundle that we use to carry out today's demonstration. Next, I'll walk you through the process for taxonomic microbiome profiling using, in this case, 16S data. And then I will hand over to my colleague, Andreas Stant, who will walk you through functional profiling of metagenomic data. We'll conclude with questions and answers if there's time at the end of the presentation. I'll begin with an introduction of the Microbial Genomics Pro Suite. The Microbial Genomics Pro Suite is a bundle of different products that's based on the industry standard for next generation sequencing analysis, the CLC Genomics Workbench. On top of the CLC Genomics Workbench, we've created a layer of specialization, or I should say maybe around. We have created a layer of specialization that provides tools and functionality as needed by modern microbial genomicists. This layer can be obtained through 3 commercial add-ons called the CLC Microbial Genomics module, the CLC Genome Finishing module, and the MetaGenMark plugin. These extensions provide functionality for microbiome profiling, both functional and taxonomic profiling, but also tools to facilitate comparative analytics. For example, to study the differential abundance in the context of metadata and really understand how changes in microbial communities are related to the biology of the underlying samples. Also included are tools for genome as well as metagenome assembly, and tools we're not going to cover today for the typing of isolate genomes. We focus on microbiome profiling today, which can be used to explore the taxonomic and functional composition of microbial communities, with the idea to really reveal keystone species that can be used as biomarkers that are, for instance, indicative of patient health or changing yields of agricultural crops or livestock, or the emergence of public health threats. The tools included for taxonomic profiling at the moment cover tools that allow our users to use 16S, 18S, or ITS, or any other amplicon data, and to use whole metagenome data to carry out functional profiling. Outputs from both pipelines can then be used with the same downstream comparative analysis pipeline to really compare results between samples and carry out differential abundance analysis. We have also brought on board of our platform commercial partners such as Cosmos ID, which completes the package by providing a plugin that allows to use whole metagenome data for taxonomic profiling, and provides reference databases that allow functional profiling with a focus on antimicrobial resistance and virulence. With that, we'll dive into the presentation of the taxonomic microbiome profiling workflow using 16S data. We use an approach referred to as OTU clustering for this, and I'll walk you a little bit through what OTU clustering means for those of you who are not familiar with this. In this case, we use the 16S gene as a phylogenetic marker for the bacterial identification. And as you know, the 16S rRNA gene encodes for a ribosomal RNA. So this is not protein coding, which is very fortunate because in in variable areas of this gene, all bases can vary freely without any restriction to the position in codons that protein coding genes have. So what typically is done is a PCR will capture hypervariable regions in the 16S rRNA gene where the primers bind to highly conserved regions. The variable regions then serve essentially as a passport for individual taxa which allow their identification. The main question researchers often ask when using 16S sequencing is which microbes are present and how abundant are they? The process to obtain them, as I mentioned, to obtain this answer is, as I mentioned, to PCR amplify hypervariable regions in the 16S gene, carry out NGS sequencing, for example, of 2 or more hypervariable regions, then assign taxonomy to the reads and count. Due to a lack of taxonomical knowledge and sequencing errors, what we do is we cluster reads at some level of similarity. So essentially what happens is the reads are clustered, as I mentioned, based on similarity, and the assumption is made that reads that are fairly similar, they represent a quasi-species. Bioinformaticians often refer to operational taxonomic unit for quasi-species. These operational taxonomic units, or OTUs, can then be reported in abundance tables. Another question that then often arises as a consequence of having the first questions answered is how many species are there? So how diverse is my sample? And how does diversity change, for example, with patient or environmental condition? Often this is also investigated to understand, was my sampling depth okay, or do I need to sequence more to capture the full diversity of my sample? Another secondary question would be, how different or similar are my different samples? So this is addressed to beta diversity or between species, between sample diversity. And lastly, often the comparative analysis is carried out to really understand which microbes are statistically differential abundant between samples or sample groups, and therefore convey biological meaning. The case we're going to tackle today is a forensics case that we've carried out in collaboration with a European Union funded project called MySafe and the Danish law enforcement. As you know, or as you may know, Denmark has a lot of medieval burial sites and Bronze Age burial sites, and it is illegal to simply excavate them as the artifacts are considered public goods. It still happens, however, that these sites are sacked, and in this case the investigators wanted to use the soil microbiome identified at the scene of the crime, as well as the soil microbiomes obtained from 2 suspects or from the boots of 2 suspects to identify whether any of the suspects can be associated to the scene of the crime. Both suspects were interviewed after obtaining soil from their boots, and both claimed, interestingly, to have been at completely different sites, not at the scene of the crime. So the investigators also sampled the soil from the locations that the suspects claimed to have been at the time of the crime. What we'll do, we'll apply 16S sequencing and analyze the data in the Microbial Genomics Pro Suite in a moment. I'll just quickly walk you through some choices that you have for the beta diversity and alpha diversity estimation. So we provide you different choices, and I'll walk through them as I present you with the workflows. But here you see that for that we provide beta diversity measures that are of phylogenetic nature, but also Bray-Curtis, Jacquard, and Euclidean diversity measures. Jacquard is a very robust diversity measure, especially with heterogeneous data, and is the least sensitive to outliers. However, more and more of our users use phylogenetic diversity measures, such as unifrac distances, often generalized unifrac distances, which offer a very robust trade-off between the unweighted unifrac distance, which gives comparatively more importance to rare lineages, and weighted unifrac distances, which gives more importance to abundant lineages. Here is an outline of the different steps that we walk through during the presentation. First, we prepare the analysis. Preparation includes the import of NGS data, the import of metadata. We'll import the 16S sequencing primer, and I'll show you how to obtain OTU taxonomy, OTU reference databases. We'll follow then with QC, with data QC and OTU clustering, alpha and beta diversity analysis, and the analysis of potential keystone or a taxa or indicator taxa through differential abundance analysis. With that, I'm going to switch to live demo and walk you through the 4 steps that are involved in preparing the analysis. What you see here is the CLC Genomics Workbench, which, as I mentioned, is the core component of the Microbial Genomics Pro Suite. The additional plugins that complete the Genomics Workbench to the Microbial Genomics Pro Suite can be obtained under this plugins menu, and they're very easy to install. Once they're installed, you can see down here in the toolbox a range of different omics applications that are supported, including the functionalities provided by the Microbial Genomics module, including OTU clustering. So the first step is to import the NGS data. So I'll navigate using the standard import function in this case to the location of the data on my desktop. I'll select the data here, which I already have done beside in that folder. I select the data, I click Next, I choose a location in my navigation area. I can create a new folder and click Finish, which will conclude the import. As the process is running, you can see down here as you switch to the process window and what is the status of a given process. Import is so fast that of course it went straight to 100%. We can also verify that the data I've imported, and yes, we can see our NGS data are here. But we require more information. We, for example, need to import also the sequences of the 16S primer so we can remove them from the sequencing data. I'll import my 16S primers into the same location, and as you see, there appear the 16S primers. It is also possible now to download a variety of different OTU databases. To download OTU databases, you could simply double-click here on the Download OTU Reference Database tool, or use the Quick Launch button in case you're not sure where the tool is located in the toolbox. In this case, you could, for example, type OTU reference, and you see the choice of available tools will appear. You can launch the tool also by simply double-clicking on the respective field in the Quick Launch menu. There's different choices available. You can use the OTU reference database provided by Green Genes, as well as Unite or Silva. I have already concluded the download, so I will not proceed with this any further. Another important step to do before you continue with the analysis is to import any metadata. So any information related with this forensics case would be helpful. In this case, it is a very simple spreadsheet that we've created that contains the geographic information of the soil that was sampled. So with that, you click to New or Import, choose Import Metadata, and navigate to the location of the spreadsheet. You can then open the spreadsheet, and in order to associate it with the NGS data, you navigate to the NGS data that we have just imported. Select the NGS data, click OK, and choose Partial Match. What Partial Match does is it looks for it compares the first column in the spreadsheet, which contains the sample name, and looks if the sample name is contained partially in the name of the NGS data sets. And since the names match, the sequencing data and the metadata here, in this case the geographic location that the samples were obtained from, are associated. As you hit Next, the last step that you have to carry out is to Finish, which will open the metadata table. This is a table that associates now all the samples in blue with the information that we have for those samples. It is very easy to provide additional information in the form of further columns, or to add further rows if you have more samples that you would like to add to this metadata table. I'll close the metadata table now, and we'll proceed next with the analysis. The analysis workflow is pre-configured and makes it very easy for you, without any knowledge about the bioinformatics that underlie the workflows, to carry out quality control, as well as the process we refer to as OTU clustering. This process includes trimming of NGS reads for low quality. It's also important to remove 16S primers here, because the 16S primers do not necessarily convey any meaningful information for taxonomy assignment. In case of paired read data, you can merge overlapping paired reads. Next, the sample is analyzed for the distribution of reads in a given sample, and outliers can be filtered out. And lastly, we use OTU clustering to produce normalized abundances and detect and filter chimeras. And I'll show you how that can be done. Again, we take a look at our toolbox, and under OTU clustering, you see there is a subfolder called workflows. Now, under workflows, there is a workflow called data QC and OTU clustering. To start this workflow, you simply double-click on it, proceed to next, and choose your input data. You can select the input data, proceed to next. At the next stage, we are asked to input the 16S primers associated to this experiment. So we navigate to the navigation area. In this case, I've done that already. We need the primers only once. And proceed to next. The default parameters that are set here will also be used towards trimming low-quality regions identified in the reads. The next step in the workflow, as I mentioned, will carry out the merging, the optional merging of paired read data. Again, I'd recommend you to use the default parameters if you're interested in what the different parameter settings mean and how they affect your analysis outcome. The answer is just one click away. Under that question mark button, you can always navigate straight to the manual, which contains this information in great detail. If you choose to change the parameters and try out different settings, and you forget what the parameter settings are that QIAGEN recommends, you can revert quickly to the default settings by clicking on this little arrow button here at the bottom. Fixed length trimming is another necessary step we carry out, and then we continue to the OTU clustering. There's two options. You can carry out de novo clustering, which will simply cluster based on similarity and not carry out taxonomic assignment, or you can choose to carry out reference-based OTU clustering, which continues with a taxonomic assignment based on the content in the reference databases that you have selected. In this case, if you choose reference-based OTU clustering, I have selected the Green Genes database, which is the output. I'll show you how to select that, which is the output of the tool that I've showed you earlier, the tool that you use to download the OTU taxonomy databases. So in this case, I'll navigate to my reference data, and you can see here for the various OTU databases such as Green Genes, Silver, and Unite, you find the content of taxonomic assignments to OTUs that have been clustered at different similarity levels. In this case, we have chosen the database part that has been clustered at a similarity of 97%, since this is what we're going to do in this analysis. You can also see here, consequentially, the taxonomy has been, the similarity percentage has been set to 97%. The setup of the workflow is complete at this stage. All you have to do is to choose an output location. In this case, we could, for example, could create a new folder and hit finish. While the workflow is running, we'll take the time and show a couple of more features of the Genomics Workbench interface. I mentioned earlier there's a process window here. The process window allows you to quickly check where you are in a process, or to terminate it in case you choose not to continue with the process. There's also ways to investigate workflows and modify the workflows according to your liking, if you, for example, have a need for greater customization. Let me show you how that is done. To that end, you navigate to the respective workflow, you right-click on it and choose open a copy of workflow. This is a graphical representation of the different tools and the order in which they're executed as part of this workflow. It is very easy to modify a workflow. Each box in this workflow represents a tool in our toolbox. The upper two portions represent an input. The middle portion represents the parameter settings that we've just configured. You can change them here. You can lock them down. If they're locked down, they're no longer configurable when the workflow is executed. If they're unlocked, they can be configured as a workflow is executed. The lower bars or lower boxes represent potential outputs. If you would like an output to be produced, you can right-click on it and choose Use Workflow as an output, which will produce, in this case, this blue box, indicating that the discarded sequences would be produced as a potential output. It is also very easy to incorporate other tools into a workflow simply by dragging and dropping tools from the toolbox into the workflow editor window. These tools can then be connected by drawing a edge or an arrow, for example, from the output of the previous tool to the input of the next tool that you'd like to run. Once you're satisfied with the workflow, you could save it and execute it just like a workflow here from the toolbox. Okay, in this case we will check on our process. The analysis has completed. Let's take a quick look at the results. So you see we've generated various reports that will tell you how many reads were trimmed for quality, but importantly, the most important output is the OTU table. This is in its most basic instance, a simple table that shows you, let's create more screen real estate here, all the OTUs that have been identified. It shows you the taxonomy that was assigned to each OTU in the order of kingdom, phylum, class, order, family, genus, and species. It shows you also the combined abundance for all the samples, as well as the abundance obtained for the individual sites. You see there's 3 sites that occur in replicates, as well as 2 suspects, GTA, which occurs in replicates, as well as GTB. This is, as I mentioned, just a very basic numeric table, but there's many ways to slice and dice and filter this further. So instead of counts, you can choose to show relative abundance, and you can also choose to aggregate, for example, at the order level. Now, bacteria have the different OTUs have been clustered and condensed to the level of the order. You could aggregate further, for example, at the phylum level, showing you relative abundances only at the phylum level. It is also possible to aggregate further based on the metadata that we have imported. In this case, we called that column type, and what type really conveys was the origin of the soil sample, so whether it came from one of the 3 sites or from one of the 2 suspects. And then you can see the combined relative abundance as well, the individual relative abundances for the individual sample groups, so the 3 sites as well as the 2 suspects. In this case, condensed to the phylum level. Much more interesting than this table, I suspect, will be the visual analysis of such data. This can be done in various different ways. Again, at the most uncondensed way, you have a series of bars, stacked bars next to each other. These bars, again, can be aggregated, for example, based on the metadata. So you could, for instance, aggregate the bars such that the abundance values from the replicates from the different sites and the different suspects occur in one bar. There are more features to navigate this visualization. For example, you could choose to aggregate the individual OTUs at the phylum level. You could also choose that the samples are sorted. At different levels. Another visualization I quickly would like to show you, which is not relevant to this case, are the so-called sunburst plots. The sunburst plots are great to investigate in depth an individual sample and the makeup of that microbial community contained in that sample. You can analyze the microbial community composition at various taxonomic levels, choosing the number of levels at that setting, going from kingdom, in this case, all the way down to family. If you would like to investigate any phylum, in this case the firmicutes, at more depth, you simply expand that view by clicking on that, on a particular wedge in that chart. You can zoom out again by clicking on the middle. Now back to the original case. Remember, the case that we're dealing with here was the investigation of a crime scene. The investigators were interested in understanding whether there are similarities in the microbial communities between the suspects and the scenes of the crime. Sorry, I wanted to stay at file level. So upon visual investigation, which is just very superficial, you might glean that the most similar samples are in fact suspect GTA and site 1. And it's important to know that site 1 was the scene of the crime. However, that is very non-quantitative, which is why in this case it's important to carry out further analysis. So the next step that we carry out in this demonstration is run a second workflow to reveal alpha and beta diversity. Alpha diversity will reveal whether we have sequenced deep enough to really fully cover the diversity contained within an individual sample. And importantly, the beta diversity estimation will allow us to actually group the samples in a statistical, more meaningful way based on the similarity in the microbiota composition. So we'll carry out that analysis next. To do that, we once again navigate in the toolbox to the workflows and choose the estimate alpha and beta diversity workflow. The input for this workflow is the OTU abundance table, which is already selected in this case, as it is highlighted here in the navigation area. Proceed with next. And here is the option to choose different diversity measures. One diversity measure for alpha diversity could simply be the count of the number of OTUs that we have observed. But there is also Shannon entropy, which is a common diversity measure, or a very meaningful measure in this case is the phylogenetic diversity. Here are the beta diversity measures that I've introduced earlier in the presentation, and I've selected Jacquard as well as weighted unifrac and the D05 unifrac distance, the generalized unifrac distance. These are all the settings that you really have to worry about. The next step is to find an output location for your results. Select this location and click Finish. Again, you can see here or under Processes the status of the analysis as the analysis is progressing. There are several tricks that I've now used during the presentation without going further into detail, so I'll take that opportunity here and show you quickly how to play with the screen real estate that you have here. For example, it's very easy to increase the viewing area in the center by reducing the left-hand navigation area, toolbox and process panel. You simply click on that button to do that. On the right side of every result, you'll always find a context-sensitive side panel. You use that to adjust the visualization to your liking and to take a deeper dive into the results. But if you need even more screen real estate, you have also the opportunity to reduce that, that window. Now we can see the process has completed. So we switch back to our navigation area and investigate the results. So let's take a look at the beta diversity estimation. To visualize that, we've chosen a process called principal coordinate analysis. And what the plot shows here is essentially each sample that we've analyzed in the form of a small sphere with a different color. Again, metadata come in really, really handy here, as the sphere color can be adjusted based on the metadata. It's also possible to hide the labels to get even more clarity in the visualization. And as you investigate this further, remember what we've seen in the bar charts where we suspected that suspect GTA and site 1 look suspiciously similar. This is confirmed by the principal coordinate analysis, where the microbiota similarity, or where the microbiota between those two samples, site 1 and suspect GTA, is highly similar, and as a result they cluster together in this principal coordinate analysis plot. Now, any judge versed in bioinformatics, or any attorney of the suspect, I suppose, might question whether we have sequenced deeply enough. And this is where this is one application that for the alpha diversity plots that we've just generated. So if you look at the alpha diversity plots, what they will tell you is well, if you look at the number of OTUs, we see the absolute number, the count of OTUs that we've obtained, or we would obtain as we would sequence deeper. You see a similar representation in the phylogenetic diversity, where a rarefaction curve eventually plateaus as we're adding more and more sequencing depth to answer that particular question. It is also possible to show the legends to really compare the different samples to each other. But what we can see here is we can see that the curves plateau nicely, which means we've sequenced at a sufficient depth for that particular analysis. So this means really it's a very clear-cut case. We've sequenced deep enough, and we understand based on that plot very well that there is a similarity in the soil in the composition of the soil microbiomes obtained from suspect 1 and the suspect GTA in the scene of the crime. The next step in this process, if this wasn't a forensics case, but if that was a microbial ecology study, would be maybe to identify which taxa are most important mostly important in their contribution to the difference between different sample groups. For example, samples that cluster here, or samples that cluster here. And this can be carried out through analysis that we refer to as differential abundance analysis. And I'll quickly show you that there are tools for this available as well in our toolbox. Under general tools, you find tools to carry out differential abundance analysis. I will not walk through this at this stage, as I, at this point, would like to switch over to my colleague Andreas, who's going to walk you through the functional profiling, because going up to the profile is different, whether you, if you want to obtain a taxonomic profile or a functional profile. But the downstream differential abundance analysis is the same, and Andreas will walk you through that. So with that, I'm going to turn over the presentation to my colleague Andreas.
Thank you. I hope I'm coming through loud and clearly. I had through a couple of hiccups during Arne's presentation, so please let me know if I'm not being clear, or yeah. So yeah, let me just switch to my presentation here. There. Okay. Yeah. So functional profiling. Yeah, so I basically stole this slide from Arne to just highlight how similar the stuff we're doing here is to what Arne just showed us. So the main question here is which functions are coded for in my sample, and here we're talking whole metagenome shotgun sequencing samples. So not just amplicon sequencing, but whole metagenome sequencing. So which functions are coded for in my sample, and we're looking at specific genes or high-level functional groups, and how abundant are these functions, and can we tell samples apart based on these abundances? And in order to look at that, we will do de novo assembly of whole metagenome datasets. We'll then call coding sequences in those assembled metagenomes and assign functions to the coding sequences. And after that, we'll build functional abundance profiles, and that will then result in a functional abundance table that looks exactly like the taxonomic abundance tables that Arne just showed you. So again, going from raw sequences to abundance profiles, and then further downstream analysis or secondary analysis could be alpha diversity, so how many functional entities do I have in my samples, and how do different samples compare with regard to internal diversity. And as I'm just talked about, is sampling depth okay? And then beta diversity, as I also talked about, how different are my samples from each other and how do they cluster? And then further comparative analysis, so differential abundance analysis, can be used to reveal functions that differ with statistical significance across samples or across groups of samples. So we'll go through all these steps in a live demo in a moment. But before doing that, if I can just change my slide, I will talk a bit more about the workflow that we use, and I'll talk a bit about benchmarks that we've done on this workflow. So this is just what I briefly mentioned before, that our workflow input here is raw metagenome sequencing reads. And the first thing we do is to de novo assemble the metagenome into contexts that originate from the same species or the same organism. Then we take those contexts and annotate them with coding sequences using a third-party plugin that is also part of the Microbial Genomics Pro Suite, namely the MetaGenMark plugin, which is the most recognized metagenome gene annotation tool. Then we take those genes and annotate with function, and in order to do that, we can either annotate it using blast hits, so using any DNA blast database you would like, or we could annotate it with PFAM domains using the standard PFAM database. Now that's all fine. So now we have our metagenome context annotated with genes and function, and then all we're missing is the abundance of each of those functions that we found. So now we map the reads, the original metagenome reads, back to our context or back to the functions that we found, and finally we build a functional profile. And so in the end we get a PFAM table showing the abundance of each of the PFAM domains we found. We get a Go table showing the abundance of each gene ontology term that we found, and we get a blast hit table showing the abundance of each blast hit we found in our context. Good. So so that's all fine, but does it work in practice? So we spent quite a lot of time on benchmarking this approach since it's a new approach, and it's a really hard problem. It's also really hard to see if it works, since no one knows what functions are really represented in metagenomes. But there's a really nice paper that came out in the beginning of last year by Stevens Lindgren and others, where they benchmarked 14 different microbiome toolkits on synthetic data. It says synthetic data here, but it's not really synthetic data. What they did is they sequenced a mock community, and then based on the reads they got from that sequencing run, they generated an error profile, and then they took all the genomes that they knew were in that mock community, and then simulated reads based on their error model. So it's sort of a mix of synthetic data and real data in this way. They built 6 data sets from 2 different communities, so 3 replicates for each community this way. And then they enriched different functions in these communities. And the way they did that was to enrich dataset A for a set of species that are known to be involved in photosynthesis and nitrogen fixation, and they enriched dataset B for a set of known pathogens. So those are the different functional categories they looked at in this paper. And then they benchmarked these 14 different toolkits, and they found that only 5 of the toolkits allowed analysis of the functional content of microbiomes, and out of those 5, only 2 of the toolkits could reliably detect the known differences between dataset A and B. So we thought this was a very nice benchmarking dataset that we would benchmark our solution on. So we went forth and did that, and this is the results from running the pipeline that I showed you just 2 minutes ago. And here you can see, so the dark blue here is the actual difference between community A and community B. So you can see it's enriched for, so community A is enriched for photosynthesis, nitrogen fixation, and community B is enriched for pathogenesis. And what you can also see here is that our solution named QIAGEN here predicts the differences in the right direction in all 3 cases. You can see it here. But not only does it predict it in the right direction, it also predicted with the sort of the right absolute fold change, or the most accurate fold change among these 6 tools, or these 6 toolkits. And the ones I marked with a star here are the ones that predict the right direction with statistical significance in all 3 cases. So those are our solution, and it's EBI, and it's MGREST. Good. So I think that the reason this this pipeline here works so well, as we've just seen, is really because of the metagenome assembler that we developed specifically for this project, or for this functionality. And we did some more benchmarks on the metagenome assembler, and they're summarized here. And this is quite a busy slide, but what you can see is that our metagenome assembler, so our metagenome assembler can be run in 2 modes. It can be run in fast mode, and it can be run in a mode where you force it to produce longer contigs, but then it takes longer to run it, and the quality goes a bit down. But if we run it in this longer contigs mode, you can force it to give very long contigs, also compared to the main competitors in this area. And you can see in all cases the quality of the contigs that are produced by our assembler is second to none. And I think this is really what makes it possible to, first of all, call coding sequences, and second of all, annotate the coding sequences that we find with reliable functions. So the assembler here is really key to the performance of this pipeline. And then, so this slide is just summarizing compute resource requirements for the metagenome assembler, and what you can see here is that our metagenome assembler basically runs on a laptop on any data set that you give it, which is not the case for the competitor sequences, and it also runs reasonably fast, as you can see here. Good. So that's all fine. That was on benchmark dataset that were synthetic in some ways at least. But does it also work on real data? So I set out to sort of test it on a manageable real dataset. This is a dataset from a paper by Matthias Willman, where they looked at the consequences of using antibiotics on the gut metagenome. So they look at the gut metagenome from 2 healthy male volunteers, subject number 1 and subject number 2. They were both treated with ciprofloxacin for 6 days, and stool samples were collected on day 0, so before treatment, so this is the baseline, and then on day 1, 3, and 6 of the treatment, and then 2 days after treatment, and 28 days after treatment. And in total, they collected 12 samples and 82.9 gigabase pairs. I think there's a typo somewhere here. Yeah. So what did they find in that paper, and what would we like to find using our tools? So they find, so they look at the taxonomic content of these gut microbiomes, and they find that gut species diversity was profoundly disturbed over the course of ciprofloxacin exposure in subject number 1, and the lowest species diversity occurred at the last day and in the following 2 days. So you can see that here. So subject number 1 is the blue here, and you can see it clearly goes down during treatment and then it goes up again, which is what it says here. It recovered almost fully after 28 days after treatment. And then subject number 2 was generally much less affected. So you can see that here it just goes up a bit and it goes down a bit and then it goes up again and stays flat. And diversity remained almost the same in all samples. So yeah, it doesn't fluctuate much like for subject number 1, which decreased significantly. It just goes up and down a bit. And finally, the values were comparable to those for subject number 1 under treatment, which you can see here. And you can also see in this PCA plot here, you can see, so 0 here is the baseline. Then on the first day of treatment, it moves, and on the 3rd day of treatment, it goes here, and then it goes all the way over here. And 2 days after treatment, it's way, well, very long away from the baseline. But then it kind of returns a bit on the way to the baseline 28 days after treatment. And subject number 2 sort of follows the same pattern, just doesn't move as much, and it returns almost completely to its baseline. Okay, so I thought, so this was an interesting data set, and it was an interesting study based on the taxonomic content of the gut microbiome. So I thought it would be interesting to see if we could get some of the same conclusions from doing functional profiling of this data set. So that's what I set out to do, and that I'll just shift to my genomics workbench and do a live demo of that. So this data set is available on SRA, so you can download it using our SRA downloader. If you go up here in the download folder and choose the search for reads in SRA, and you just type in the project number, so PRJEB10391, and hit enter. And then you wait for a bit, and then you get so this is basically all the 12 samples, so all the samples from subject number 1 and all the samples from subject number 2. Now you can just highlight all these, press Ctrl A, and say download reads and data. And then you can run it in your workbench, and you have different options here where you can, if you want to decrease the amount of data, so the raw amount of data would be 160 gigabytes of disk space, so you can decrease the amount of data by discarding quality scores or read names and get it down to 90 gigabytes of data. I'm not going to download that now because it would take hours. But what you get when you download it is up here somewhere. You get, yeah, so here's all the data. You get all the data and you get a metadata table. I'm not going to work directly on this data because it takes a long time to assemble, or it takes a fairly long time to assemble metagenomes. So I'll go directly to show you the metadata that we also get. So this is the metadata table that we get when downloading the data, and then I just added a couple of columns here. So I added this day column here, and I added this treatment column here. Otherwise, it's exactly the metadata table we got from the SIA downloader. Then the workflow I just showed you is this one. So we trim the sequences, we de novo assemble it, we annotate it with coding sequences, we annotate it with PFAM domains and Go terms, we map the reads back to the context, and then we build a functional profile based on that. So that's the workflow. In order to run that workflow, we need two databases. We need a PFAM database, as you can see here, and we need a Go database, or gene ontology database. We have tools in the workbench to download those. So I click the launch button up here. I type PFAM, and I can find the download PFAM database. If I type Go, I can find the download Go database. I also already did that. So that's what you saw here. Here I supply the PFAM database, here I supply the Go database, and I can pick a slim set of the Go terms. So there's the complete Go basic, which contains all the different Go terms in the complete Go, and then there's sort of different subset or different slices of the Go ontology, gene ontology. I used this metagenomics Go Slim for this analysis. So then, in order to run it, I pick my samples here in the right. I highlight all of them. I run. I click next, and then they're all marked here, and I just transfer them to this area, and then I step through. And in the final, here I choose to save it, and then I can find a place to save it. Now I'm not going to run it because it takes too long for this demo, but I can show you the output of running it, which I saved down here. So again, I get a PFAM table and I get a Go table, and then I could have gotten a blast table, but I didn't annotate with blast hits, so I don't get that in this case. The PFAM table looks like this. It looks very similar to the OTU table that Arne just showed you. It has the name of the PFAM domain, what clan it is in, and then the combined abundance if I had more than one sample in this table, and the abundance for this specific sample. And it has all the different views that Arne also showed you, the stacked bar chart view and the sunburst chart. Then the next step now is, so now I have a PFAM table for each of these 12 samples, so I'm going to merge those using a tool called merge abundance tables. So I click my launch button up here, type merge. I choose the merge abundance tables tool. I click next, and then I could click through here and say, okay, I want this PFAM table here, and go do that for all 12 samples. But what I'm going to do is just to say, to right click on this folder here and say add folder content recursively. And then it automatically picks the 12 PFAM tables for me. Click next, and I choose to save it. And I save it down here in my folder and hit finish. And that was really fast. So now I have my merge table here, and now I have a column for each of the 12 samples, and a row for each of the PFAM domains that were found in any of these samples. I'll close this, and now the next steps are again alpha and beta diversity. So I'll start with alpha diversity because that takes the longest to run. So I run the alpha diversity tool, click next, select my table, click next again, and then I'll just use this Chow1 bias corrected alpha diversity measure. I could use any of these 5, but this is the one that separates the data the most. So this is the one I'm going to demo here because it's nice for visualization purposes. I just keep the other parameters as they are per default. Click next, and then I will save it. I'll save it down here in my alpha diversity folder. And now while this is running, I'll also just do beta diversity. Oops. I run the beta diversity tool. I click next. I choose my table, and then I'll use this Bray-Curtis. So this is the one that's most robust to noisy data, I would say. I could also use these two, but yeah, the Euclidean is really sort of, if you want to tell stuff apart that's really close together, you could use the Euclidean. Otherwise, I would use one of these two. Sorry. Click next, and here I just choose to open the output. I click finish, and it opens. So now I can lay out my my principal coordinate analysis plot here. This is already done automatically because I've run this this analysis before, and I saved it down here. So you can save your view settings either for this particular object itself, or for this PicoA 3D viewer in general. And then when you want to apply it, you just say, okay, I want to apply this this setting that I saved. Sorry. Sorry about that. Okay. So here, what you see here is the purple dots here are sample number 1, and the orange dots are sample number 2. So you can see a clear separation of the two subjects. You can also see. So I chose to show the label text as the day of treatment here, and you can see the baseline for sample number 1, or for subject number 1, sorry, is here. On the first day of treatment, it moves here, and then during treatment, it moves down here 2 days after treatment, it's all the way down here, and it gets all the way back to the baseline on day 28 after treatment. And you can see the same pattern for subject number 2. And now I think the alpha diversity finished. We can have a look at that. And here again, I'll do that trick and apply the settings I saved. And now it looks like this. And here you can see, if we just zoom a bit on the last part of this plot, you can see that subject number 1, which is the full lines, started with very high diversity. During treatment, it went all the way down here to the bottom, and then it went up again and finishes right here with the red line. Subject number 2 starts with lower diversity, and it goes up a bit and then down, and then it finishes here at its baseline again. Okay, so with that, I'll finish the live demonstration, and I think we're also up against the hour, so I will hand over the presenter rights to Arne again for some final comments.
All right, thanks Andreas for the presentation. As Andreas mentioned, we're up against the hour, and normally we would use the time for Q&A session. So I'll ask our moderator Miranda if we have any received any questions, and maybe we'll have a few minutes to answer them. And while Miranda looks into the questions that we've received, just a quick note or a reminder to all of you to visit QIAGENbioinformatics.com, where you can not only receive a free trial, but also all the instructions on how to install the trial and obtain data and information and tutorials to really get the most out of testing the software. You'll find the information on the Microbial Genomics Pro Suite by going to QIAGENbioinformatics.com. Under Solutions, you'll find Microbial Genomics, which will give you access to a wealth of resources. And of course, as soon as you encounter any trouble, please don't hesitate to reach out to us. You can write us via email to support-clcbio@qiagen.com.